Hi, I'm David and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. Figure 1.1 shows the cell surface membrane. It typically measures around 5 to 10 nanometers, with 7 as the most common width. A channel protein is an integral membrane protein that forms a hydrophilic pore through the cell surface membrane. It allows water, ions, and other polar substances to pass through via facilitated diffusion. You can get this marked by stating any one of these categories. A in the diagram that looks like a tunnel is a channel protein. E, the phospholipid, is the main component of the membrane. It forms a bilayer, with its fatty acid tails facing each other. The tails form a hydrophobic core, which act as a barrier to water-soluble or polar substances. Fat-soluble and non-polar substances are allowed to diffuse through. Phospholipid molecules can move freely within their monolayer. This gives rise to the fluidity of the membrane. Receptors on the cell surface membrane are usually glycolipid or glycoprotein. B is a glycoprotein. It is composed of a protein bonded to a carbohydrate chain. D shows a lipid molecule that has carbohydrates attached to it. It is a glycolipid. Cholesterol features a 4-ring structure consisting of 3 6-membered rings and 1 5-membered ring. It gives stability and maintains fluidity of the membrane. At low temperatures, it prevents the close packing of the fatty acid tails, increasing fluidity. This is because they reduce the intermolecular interactions between the tails. At high temperatures, it stabilizes the membrane by preventing it from becoming too fluid. They insert their rigid ring structure between phospholipids, restricting their movement and preventing excessive fluidity. It forms the hydrophobic core together with the fatty acid tail of phospholipids, so it can also act as a barrier to the water-soluble substances. B wants you to draw and label a few organelles. Mitochondrion is a double membrane structure, with the inner membrane folded into cristae. The rough ER is composed of interconnected flattened sacs called the cisternae. The smooth ER is a tubular structure with no ribosomes. X is a Golgi body. It consists of a stack of flattened cisternae and associated vesicles. It modifies and processes biological molecules such as proteins and lipids. For example, the addition of carbohydrates to protein to form a glycoprotein. This process is called glycosylation. It also packages proteins into vesicles to transport them to other parts of the cell or out of the cell via exocytosis. There is a mark for AVP. You can talk about the formation of lysosomes or how they are involved in cell wall formation. 2A wants you to explain how the properties of water make it suitable as the main component of blood. This question wants you to focus on the blood. So, do not discuss the properties of water related to transportation in plants or their role in the ecosystem. Water is known as the universal solvent. It can dissolve ions and polar molecules, allowing them to be transported in the bloodstream. You can get the second mark by stating the transport of a named substance for a correct reason. Water has a high specific heat capacity. It is the amount of heat that must be added to 1 gram of water to increase the temperature by 1 degree Celsius. It means a large quantity of heat is required to increase the temperature of the blood. So, the temperature of blood remains fairly constant in our body despite the changes in the ambient temperature. Its great ability to disperse heat throughout the body allows different parts of the body to have similar temperatures. There is a mark for AVP. You can discuss the dipolar features of water molecules and how this can help to dissolve ionic compounds for transportation. Q delivers blood to the right atrium. It is the vena cava. P is the aorta. It carries blood from the left ventricle to the rest of the body. 2. Describe the functions of P and Q. The aorta transports oxygenated blood to the body. Oxygen and respiratory substrates such as glucose are delivered to the respiring tissues. It is the main artery of systemic circulation. Systemic circulation is the circuit of vessels supplying oxygenated blood to and returning deoxygenated blood from the tissue of the body. The blood is transported at high pressure to reach the organs. Vena cava is the largest vein. It returns deoxygenated blood to the heart. The blood has a low pressure and is delivered to the right atrium. 3 wants you to explain the closed double circulation. In this type of question, you should explain both words separately. Closed refers to the fact that blood is contained within the blood vessels, making it a closed system. Double refers to the blood flow that passes through the heart twice in one complete circuit. It must complete the pulmonary and systemic circulation in one full circuit. You can also describe the circulation to get this mark. Figure 2.2 shows the cross-section of an arteriole. You can see a few red blood cells in the lumen. 1. Explain why it is important that the blood pressure decreases as it passes through arterioles. Arterioles deliver blood to the capillaries. If the blood pressure remains high, capillaries may burst. Capillaries are the smallest blood vessels that we have, with very thin walls that are composed of only one layer of endothelial cells. It is not made to withstand high blood pressure. Another reason is to slow down the blood flow. Material exchange occurs near the capillaries. Slower blood flow allows more time for the diffusion of substances, increasing efficiency. There is a mark for AVP. The low pressure can effectively cause an even lower pressure in the venous end of capillaries. Tissue fluid is reabsorbed in that region, achieving material exchange. 2. Compare the structure of a muscular artery with the structure of the arteriole. A muscular artery is a medium-sized artery that branches out from an elastic artery. It has a thick and well-developed tunica media that enables it to contract and relax, thereby regulating blood flows to specific organs and tissues. The command word here is compare. It means that you have to include both similarities and differences. Both have the 3 layers you can find in arteries. However, the thin tunica adventitia is not clearly seen in the photomicrograph, so it is not credited here. The muscular artery is a larger vessel, so it has a wider lumen. However, due to its thick wall, the ratio of wall thickness to lumen width is smaller. The thick wall is mainly due to the thick layer of smooth muscle in the tunica media. Tunica media is the middle layer of a blood vessel wall. It also has more elastic fibers to allow a greater ability to stretch and recoil. A thicker wall also means that it has more collagen fibers. This is important because the blood pressure is higher in the muscular artery, and collagen fibers enable it to better withstand the pressure. There is a mark for AVP. You can discuss the tunica adventitia here. 3a shows a flowchart to identify infectious diseases. Malaria is a vector-borne disease. It is transmitted by the female Anopheles mosquito. 4 main species of Plasmodium can cause the disease. Any one of the names can give you the mark. TB is an airborne disease, and it is transmitted by contaminated air droplets. It is caused by bacteria called Mycobacterium tuberculosis and Mycobacterium bovis. A bacterial infection that is transmitted by the fecal oral route is cholera. The pathogen that causes the disease is Vibrio cholerae. B shows us the structure of a DNA nucleotide and a HIV drug, tenofovir. It is used to interfere with the action of reverse transcriptase. You are asked to suggest the mechanism of action of the drug to prevent the synthesis of DNA. From the diagram, we can see that tenofovir have a similar shape to a part of a nucleotide. So it is likely to act as a competitive inhibitor. It binds to the active site of the reverse transcriptase due to their complementary shapes. This blocks the binding of the actual nucleotides, so they are not added to the elongating chain. The phosphate group allows the drug to form a phosphodiester bond to the elongating DNA strand. However, it has no 3' hydroxy group, so the next nucleotide cannot form a phosphodiester bond with it. This stops the reaction from continuing. C1. Calculate the percentage of people who received PrEP in 2019 as a percentage of the target set by UN in 2016. Divide the number in 2019 by the target and multiply by 100. Remember to round it to the nearest whole number before writing it down in the answer space. 2. State and explain how health authorities can reduce the transmission of HIV. You will get 1 mark for stating the strategy and another mark for its explanation. The strategies must be related to the health authorities, not something that can be done by an individual. They can supply condoms or femidoms for protection during sex. These items can act as a barrier to transmission during sexual intercourse. Needle exchange schemes can be carried out so that intravenous drug abusers can obtain clean needles. It is a service that allows injection drug users to obtain clean needles at little or no cost. This decreases the risk of them sharing contaminated equipment. In the hospital, new needles and sterilized equipment must be used for all medical procedures. This can decrease the risk of transmission from contaminated blood. Free testing can be provided for high-risk groups such as sex workers and men who have sex with men. This allows early diagnosis, so newly infected people can start their drug treatment immediately. Pregnant women should be tested. If they are HIV positive, they should not breastfeed their children, as this may be a method of transmission. Health facilities should prevent people who are HIV positive from being blood donors. They should always screen donated blood and heat treat it. This can prevent people from receiving blood infected with HIV during blood transfusions or operations. Hospitals can carry out contact tracing. It is the process of identifying and notifying the sexual partners of a person diagnosed with HIV, so they can get tested, receive care, and take precautions to prevent further transmission. Free drugs can be supplied to people living with HIV. This increases the treatment rate and helps to reduce the viral load. HIV viral load is the amount of HIV present in a person's blood, measured by the number of viral RNA copies per milliliter. Healthcare providers can help in campaigns to provide education about HIV treatments or transmission to the public. This helps to raise awareness of ways to reduce the infection. There is a mark for AVP. As long as you can provide any solid suggestion, you can get this mark. Figure 4.1 shows the tissue that lines the bronchi. The layer nearest to the lumen is the ciliated epithelium. It is composed of two types of cells, ciliated epithelial cells and goblet cells. A is a columnar-shaped cell with cilia. It is the ciliated epithelial cell. B has no cilia, and you can see the mucus being secreted. This is the goblet cell. 2. Describe how the tissue shown in figure 4.1 is adapted to its function in the gas exchange system. Goblet cells are specialized to secrete mucus. Mucus is composed of glycoproteins, specifically a glycosylated protein called mucins. Mucus covers the epithelium to trap particles. This prevents pathogens from reaching the epithelial cells or moving further down to the alveoli. It is a defense mechanism to reduce the risk of lung infections. Cilia in the bronchi move by performing a synchronized wave-like beating motion powered by their internal microtubule structure. They can move mucus and the trapped materials upwards towards the mouth away from the alveoli. Eventually, we can move these unwanted substances out of the airways. Figure 4.2 shows the cross-section of the cilia. You are asked to describe its internal appearance. Cilia are composed of microtubules. All those circles you see in each cilium are the cross-section of the microtubule. They show a 9+2 arrangement. It means there are 9 pairs of microtubules forming the outer ring, and 2 in the center. Each pair is called a doublet. There is a mark for AVP. You can get this mark by describing the meaning of 9+2 arrangement. Intracellular means within the cell. Each cilium is surrounded by the cell surface membrane, so we know that they are considered a structure within the cell. C. Describe the function of centrioles and explain how they are involved in the cell cycle of a stem cell. Centrioles act as MTOC. MTOC stands for Microtubule Organizing Center. It is a structure within eukaryotic cells that serves as the site of microtubule organization and assembly. During the cell cycle, it organizes microtubules to form the spindle fibers. During the S phase, each centriole replicates. We will have 2 pairs afterwards. The centriole pairs move to the opposite poles of the cell during prophase of mitosis. They are arranged at right angle. Together with the protein surrounding them, they form the centrosome. Centrioles can lengthen and shorten the spindle fibers to attach them to the centromeres and to pull the sister chromatids apart. Figure 5.1 shows the effect of LVPD on the rate of transpiration in 2 different plants. A. Compare the results of the 2 species. The command word compare indicates that you have to discuss both similarities and differences. The starting point of both lines shows that there is no transpiration when LVPD is 0 kPa. Then, both increases in transpiration rate as LVPD increases until 2.5 kPa. However, helianthus rate remains constant after that. Nerium's rate decreases slightly. Overall, helianthus always has a higher rate of transpiration. It also shows a steeper gradient. Any comparative data quote from figure 5.1 will give you a mark. When quoting data, remember to place the point after the statement you are supporting. Do not put the data at the end of your answer unless it is used to support your last descriptive statement. Figure 5.3 shows the cross-section of oleander. Figure 5.4 zooms in on the epidermis. You are asked to state and explain two adaptations of the leaves. Note that your answer must be something that can be seen in the figures. Oleander is a xerophyte. These features help them to reduce excessive water loss by transpiration. In figure 5.3, we can see 3 layers of thick wall cells at the upper and lower epidermis. The extra layers are hypodermis. They increase the distance of water vapor to diffuse, reducing the rate of cuticular transpiration. Cuticular transpiration is the loss of water vapor directly through the plant's waxy cuticle. There are no stomata on the upper surface. This feature prevents the exposure of stomata to direct sunlight, as it can trigger the opening of stomata. At the lower epidermis, stomata are found in pits or crypts. Pits are the depressions or grooves found on the epidermis. This chamber allows water vapor to gather and creates a humid atmosphere to reduce the water potential gradient. It also minimizes the effect of external air currents, preventing water vapor from being carried away. In figure 5.4, we can see that a thick wall epidermis reduces the diffusion of water vapor through the lower epidermis. It reduces the rate of cuticular transpiration as well. A thick waxy cuticle can also be seen. Waxy cuticles are waterproof. It reduces the diffusion of water vapor through the cuticle as well. Epidermal hairs called trichomes are found around the stoma. They trap still air and create a humid atmosphere around the stoma. This reduces the water potential gradient, hence the rate of water vapor diffusion. Figure 6.1 shows an antibody molecule. A1. Explain how the structure of an antigen binding site makes it specific to a particular antigen. The antigen binding site is found in the variable region. It has a shape that is complementary to a specific antigen. It has a specific primary structure. In other words, there is a unique sequence of amino acids in that region. Different amino acids have different R groups, leading to a variety of R group interactions. These give rise to specific tertiary structures, hence the 3D shapes for the binding to antigens. There is a mark for AVP. You can talk about the epitopes of the antigen or name the R group interactions. The hinge region gives flexibility for its binding to antigens. The two antigen binding sites can move and bind at different angles. This ability allows them to bind to antigens that are at different distances apart. 3. Suggests an advantage of antibodies bind to receptors on macrophages. An antibody can bind to the receptor on macrophages using its constant region. They have complementary shapes. The binding makes it easy for macrophages to engulf the pathogen as they have the antibody-antigen complexes. Antibodies mark the pathogen and facilitate its destruction. In other words, antibodies act as tags that signal to macrophages and other phagocytic cells where to find and engulf these pathogens. This is called opsonization. B. Suggests how the modification of the primary transcripts occurs in plasma cells. A primary transcript is the initial RNA molecule produced directly from a DNA's template strand during transcription. It contains both coding regions, exons, and non-coding regions, introns. It must go through the post-transcriptional modification to become the mature mRNA. One of the processes is called RNA splicing. Introns are removed from the primary transcript. After that, exons are joined together. The exons can be joined in different sequences or combinations, forming mature mRNA with different nucleotide sequences. This means a gene can be used to produce polypeptides with different primary structures. This is known as alternative splicing. This is possible because not all the exons are used in making all the polypeptides. That's all for today. If you think my videos are useful,
Don't forget to like, comment, and subscribe. If you have any questions, feel free to ask me at the comment section. Thank you for watching and see you again soon.